All right, let's go over our Unit 2 test so that we can absolutely crush our retake, right? Okay, so which process uses energy to combine inorganic molecules to synthesize organic molecules? Let's see, what are some inorganic molecules? Well, we know that organic molecules must contain carbon and hydrogen, right? They have to contain both of those two. So let's see, what are some inorganic molecules that we can think of? Respiration. Let's see, we know that respiration takes glucose, which is C6, H12, O6, plus oxygen, produces CO2, and what? Water. Okay, CO2 stands for carbon dioxide. So let's see, are we synthesizing organic molecules? Synthesizing means to make. Are we making organic molecules? These are the things we start with, and these are the things we end up with. Are any of these organic? Do they contain carbon and hydrogen? Nope. This contains carbon but no hydrogen. This contains hydrogen but no carbon, so that is not correct. Digestion, this is where we take very large things and break them down into smaller particles, so we're not synthesizing anything. Photosynthesis. Let's see, what's the equation for that? We've got water, we've got carbon dioxide, and that is making, I'm just going to write out glucose. No, all right, sorry, that's really messy. C6H12O6. There we go. Plus oxygen, right? This happens in plants. They make the oxygen we breathe and the sugar that we eat. Let's see, is that using combining inorganic molecules to synthesize organic molecules? Let's see, is water inorganic? Yeah, it's inorganic because it doesn't contain carbon. Is carbon dioxide inorganic? Yeah, to be organic, it has to contain carbon and hydrogen. So if it's inorganic, it doesn't. So that means it doesn't contain hydrogen. So these are inorganic molecules. Good. Are we making organic molecules? Let's see. Are we making carbon, hydrogen? Oh, that looks like an organic molecule, right? So I'm going to go with that one. Decomposition we haven't talked about yet, but this is where bacteria or fungi break down nutrients back into their smaller forms in the environment. So I'm going to go with C. Photosynthesis uses inorganic molecules like water and carbon dioxide to make organic molecules like glucose. All right, how do I go forward? I erase all and go forward. All right, yay, it worked. Okay, question 2. Part of a laboratory procedure is shown in the diagram below. This setup would most likely be involved in a procedure to do what? Well, you guys all know this. I think a lot of people got this one right. This is where we took different solutions and we mixed them with Benedict's. And we heated them up for like a few minutes and they turned from blue to orange if they contained, this is not easy to write, but I'm just trying. All right, blue to orange. Wow, that's really bad. If they contained glucose. So stained specimens while making a wet mount. No, you guys know what that is. Separate pigments in a mixture. We haven't done that yet. Determine pH of solutions. Nope. This is what we do to measure glucose. Okay. Wait, hold on. Erase everything. All right, this is a tough one. But start by thinking about the question. Before you even look at the answers, look at the question. Okay, a biological process that occurs in plants. Okay, so we know that there are 2 things that occur in plants. Well, a lot of things that occur in plants. But we know that photosynthesis only occurs in plants and other autotrophs, like algae and all that fun stuff. So photosynthesis, this could be photosynthesis. Plants also undergo respiration. So let's see which one it is. Is it photosynthesis or is it respiration? All living things undergo respiration. So just because it's a plant, This is terrible. Look at that handwriting. Holy smokes. All right, so all living things undergo respiration. Let's see which one this is. Let's see, we're using water. If we're using water, we're taking in water. We're taking in something else, and we're producing two things. Let's go through. Let's see, if we have water and enzymes in the presence of glucose, this means like in the presence of glucose. Something that's written above the arrow is like something that helps it along, but it's not necessarily an ingredient. Let's see, if we had water plus enzymes in the presence of glucose produces oxygen and carbon dioxide. That doesn't sound right? We know that we don't really have any chemical reactions where oxygen and carbon dioxide are both produced. And enzymes are not really reactants. They more help things along. So I'm going to go no for that one. Let's try number 2. Hey, where's my pen? Okay. Carbon dioxide, water plus carbon dioxide, okay, in the presence of light and enzymes. All right, water, carbon dioxide, presence of light and enzymes. That sounds kind of like photosynthesis. Let's see if it continues. B glucose. And C, oxygen. Is that our reaction for photosynthesis? I have to find a better way to write these words. That is, water plus carbon dioxide and in the presence of light and enzymes produces glucose and oxygen. That is definitely photosynthesis, so that could be true. Now we just have to watch out to see if respiration is down here too, because respiration is also a possible answer. Respiration, we know, is the opposite of this reaction, right? Respiration takes oxygen, it goes in reverse. Respiration is oxygen plus glucose producing carbon dioxide and water. Let's see if we have that. Wait, no, we can't possibly have that, right? Because we already know water's on this side. Do any of these make sense? Water plus glucose in the presence of carbon dioxide makes enzymes and oxygen? No. Water plus oxygen in the presence of light and enzymes produces glucose and carbon dioxide. That is also an incorrect answer because we know that water plus carbon dioxide has to produce glucose and oxygen. Just remember that trees make oxygen and trees make glucose, and then everything else kind of falls into place. So the correct answer is 2. All right, some blood-sucking insects insert their mouth parts directly into a blood vessel and withdraw blood. Other blood-sucking insects have mouth parts that cut through the skin and the blood vessels and produce a small pool of blood from which they feed. Both are gross. Just kidding, it's just science, right? But both mouth parts are specialized for autotrophic nutrition. What's autotrophic nutrition? Autotrophic nutrition is when something can make its own food. Would you suck blood if you could make your own food? No, you'd undergo photosynthesis. So I'm going to say that's not true. Heterotrophic nutrition. What's heterotrophic nutrition? That's something that has to get its food from someplace else. Would you suck blood if you had to eat for a living? Yep, you probably would. Regulation, that doesn't sound like regulation. Regulation is when systems interact with each other. Excretion is getting rid of waste. We know he's not getting rid of waste, he's eating. So the correct answer is heterotrophic nutrition. This would be something that has to get its own food from another source. Can't make its own food. All right. All right, the next question. Certain organisms are able to store energy in compounds. Certain organisms are able to store energy from the sun in energy-rich compounds. Which event best illustrates this activity? First, you should just maybe think about which organisms could store energy from the sun. Well, gosh, to me, that's going to be a plant or an autotroph, right? Otherwise known as an autotroph. So let's look at which ones of these are, I just spelled autotroph wrong, T-R-O-P-H. There's got to be a better way for me to write on these. Okay, so which one of these is a plant? And by the way, what is the energy-rich compound that we're storing it in? That's glucose, right? Is a fox an autotroph? No. Is a caterpillar an autotroph? Or is a blackbird an autotroph? No. Is a lettuce an autotroph? It's a plant, right? It's green, probably makes glucose. Bacteria are not necessarily autotrophs. And particularly when you look at this, it says they're taking organic material and breaking it back down into simple nutrients. So these are taking something large and they're breaking it back down. These are called decomposers. We'll talk about that later. But this is the only autotroph on the list. So the correct answer is C. Okay. Next. Oh, you guys should all, I think a lot of people got this one right. The stomate is an opening in the bottom of a plant leaf. And it's a little opening that's protected by 2 guard cells on either side. It helps maintain homeostasis in the leaf by allowing gases to come in. For example, let's see, carbon dioxide is going to come in, right? Because that's what we need for photosynthesis, and oxygen is going to come out. We're probably going to lose some water vapor in there too. So what is it similar to? Stomate in the lower surface of the leaf has a function most similar to what cell structure? What lets things in and out of the cell? Cell membrane. Right, ribosome makes proteins, vacuole stores food and waste, and the nucleus has all the genetic instructions. All right, on to the next. Okay. So this is a Venn diagram. What are characteristics that are represented by both of these processes? Diffusion. Diffusion is when molecules move from a high concentration to a low concentration. Active transport is when molecules move from a low concentration to a high concentration. Remember the picture of the hill, the guy on the hill, going from high to low? Super easy, you just roll right down the hill, right? But if you want to go to that same hill and you want to go from low to high, that requires energy, but you're still moving. Okay, so what do they have in common? Do they both use ATP? Nope, diffusion does not use ATP because it doesn't use energy. Do they both require enzymes? No, diffusion just happens. We did a lab with glucose inside of our mock cell, and we didn't add any enzymes, and the glucose still moved. Do we use oxygen? Nope. Oxygen is required for producing ATP, so active transport uses oxygen. But do they both move molecules? Yep. Diffusion and active transport, both movement of molecules in a given area. Number 8. Okay. ATP is most likely being used for what? Well, when you see a picture like this, I always like to count the molecules. So first of all, we see, let's look at A's. We've got 1 A outside. How many A's inside? 4 A's inside. All right, let me see if I can change the color and be super fancy here. Okay, and let's look at B's. We have 1 B inside. 1, 2, 3, 4. 4 B's outside. Okay, so ATP energy, that's being used, that's active transport. So if you picture the little guy going up the hill, active transport requires energy, so to pedal up that hill, they're going from low to high. Right? So which one of these is going from low concentration to high concentration? Let's see. Substance A entering the cell. It's going from a low concentration of 1 to 4, so that could be it, right? Substance B to enter the cell. There's 4 outside and only 1 inside, so that's going from high to low, so that's not going to require energy. That's going to be diffusion, passive transport. Both substances to enter the cell. Nope. Because we already just said B doesn't need energy. Neither substance to enter the cell. Nope. Correct answer is A. You need energy to go from low to high. All right, number 9. Which row in the chart below best describes the active transport of molecule X through a cell membrane? All right, if we see these words, active transport, we know we're probably going up that hill again, so it's going to be from low to high. We also know, because you need energy to pedal that bike up the hill, we also know that it's going to require ATP energy. All right, so now we know what we know. Let's look at the answers. All right, which one shows movement of molecule X? High to low? Nope. We already said it wasn't high to low. We already said it's low to high, right? And we're using energy. So we just answered our own question. The answer is going from low concentration to high concentration, and it has to use energy. That's the definition of active transport. Number 10. All right, this problem we did in class a million times, so you guys should know this. But this it is a little tricky. So oxygen molecules entered the cell. Okay, sorry. Let me read this first. Diagram below represents the distribution of some molecules inside and outside of an artificial cell over time. We've got water, we've got oxygen, and we've got protein, right? So let's look to see what's changed between the two of them. 1, 2. Let's just look at water first. 1, 2, 3, 4, 5, 6. Water's outside. I'm going to make water blue. And I'll put it right by the water molecule. So we've got 6 waters outside. 1, 2, 3, 4, 5, 6. 6 waters inside. Okay, and then after we let it sit for a little bit, we have 1, 2, 3, 4, 5, 6. We still have 6 waters outside. 1, 2, 3, 4, 5. We still have 6 waters inside. So water didn't really change. Right, and now let's see, what color should we make oxygen? I'm gonna make it dark blue. Oxygen, 1, 2, 3, 4, 5, 6, 7 outside. 1, 2 inside. And after time 1, 2, 3, 4, 5, we have 5 outside. And 1, 2, 3, 4 inside. So oxygen changed. Now let's look at proteins. I'm gonna make proteins purple. 1, 2, 3, 4, 5, 6, 7 outside. Inside 0 outside, and then 1, 2, 3, 4, 5, 6, 7. Still 7, 0, so it didn't change. Now let's go through our answers. Oxygen molecules entered the cell over time by active transport. All right, oxygen molecules. Looks like some oxygen molecules did enter the cell, right? So we went from there was 7 outside and 2 inside, and now there's 5 outside and 4 inside. So some oxygen molecules did enter the cell, but the tricky thing is, here we're going from, to be considered active transport, we have to go from low to high, right? And we're seeing there was a high concentration outside, and it moved into a low concentration. So that would not be active transport. If this said diffusion, I would say yes, but if it's active transport, no way, because it went from a high concentration outside into the lower concentration inside. Water molecules are too large to leave the cell, so they remain where they were at the start of the investigation. Well, they did stay where they were. Water didn't change, but it's not because they're too big. They are tiny, and they flow very freely through the cell, through osmosis. Protein molecules are kept inside the cell because the cell needs them. This is where some people got tricked up. Proteins are kept inside the cell, right? But it's not because the cell necessarily needs them. The cell also needs to get rid of proteins. It's constantly shuffling proteins in and out of cells. But the problem is that they are too large to diffuse outside of the cell. They can't just diffuse. You need active transport to get these proteins out because they're so big. So the correct answer for that would be D. Number 11, function. Oh, you guys know this. Shape. Shape determines the function of an enzyme. You just have to remember that one, like a lock and a key, right? Okay, calcium concentration in the root cells of certain plants is higher than in the surrounding soil. All right, let's draw that. Calcium concentration in the root cells. Let's draw a plant cell. I'm going to make it green. All right, so here's our root cell. Okay, it might be square, cell membrane, cell wall. And we'll just leave it like that. Okay. The calcium concentration in the cells of certain plants is higher than in the surrounding soil. So let's just say we've got some calcium in the cell. It says it's higher in the cell than it is in the surrounding soil. So let's make 3 in the cell and 1 calcium ion out of the cell. Calcium may continue to enter the root cells of the plant by the process of what? All right, we have 1 molecule in, 1 molecule out, 3 molecules in. So to enter the root cells, you're going from 1 to 3, which is low to high, which is active transport, right? Because remember the hill guy? Going up the hill takes energy. Number 13. The inability of an organism to produce certain proteins can occur when an organism is lacking an enzyme needed to combine what? Well, let's see. Production of proteins. Proteins are just amino acids linked together, right? Why am I even writing that? Doesn't make any sense. Okay, but proteins are amino acids just linked together. We should know that. So if they don't have the enzyme to combine the amino acids, they're not going to make proteins. Enzymes are biological catalysts. If you can't produce proteins, simple sugars would mean it couldn't produce starch, oxygen molecules. They don't really get linked up to make anything major that I can think of. Okay, so the correct answer for that is C, amino acids. Amino acids are the building blocks of proteins. So if they don't have the enzyme to combine amino acids, they can't make proteins. Number 14, which group consists of entirely organic molecules? All right, well, let's look for carbons and hydrogens. We know proteins are cho. Remember we did cho, chon, chomp. Proteins are chon. Fats are cho. Proteins are chon. That stands for carbon, hydrogen, oxygen, nitrogen. Carbon, hydrogen, oxygen. Carbon, hydrogen, oxygen. Sorry, it's messy. Water is H2O. Okay, we can just stop there because we know already that's not organic, right? Because it doesn't have a carbon. Not organic because oxygen doesn't have a carbon. Water, it's inorganic. Inorganic means not organic. Doesn't have a carbon. So the correct answer is B. All of these molecules contain carbon and hydrogen, so they are considered organic molecules. Okay. Number 15. Cell membranes are said to be selectively permeable. That's the same as semi-permeable. Which statement best explains what selectively permeable means? It means the cell membrane can prevent any harmful substance from entering the cell. No. The cell membrane, when you see the words like any and all, they might be potential words that might trigger you to eliminate as an answer because usually things are not so absolute. The cell membrane lets certain substances enter the cell and keeps certain substances out of the cell. That sounds good. Do large molecules diffuse into the cell? No. Remember we saw the starch? It got stuck in the cell. It couldn't move at all. The cell membrane has pores that only let out water and glucose into the cell. Only let water and glucose into the cell and carbon dioxide out. Well, it does let water and glucose into the cell and carbon dioxide out. But is that only what it does? No, we've got all kinds of other things that are going in and out. We've got oxygen, we've got all kinds of different nutrients. So that's a little extreme. Look, there's another keyword, only. So the correct answer is B. Certain substances in and certain substances out. All right, number 16. In the human body, carbon monoxide, which just happened. Whoa. Sorry. Okay, carbon monoxide, which is chemically C. Whoa, why? It's all messed up here. Carbon monoxide is CO, just in case you're wondering. Reduces the amount of oxygen that can be transported to cells. Breathing in too much carbon dioxide will most likely result in the production of what? Well, first let's think about what oxygen is needed for in the cell. Cellular respiration, right? Oxygen plus glucose produces, right? It's the opposite of photosynthesis, exactly the opposite of photosynthesis. Produces carbon, except for we've also got some ATP in here. And we also lose it we also lose some heat too. So let's see. If we don't have enough oxygen, then this reaction is not going to go that smoothly. So we're going to have less of all of these things. Less carbon dioxide, less water, less ATP. Nope, not less glucose, right? That's one of the reactants. We might still have plenty of that. More DNA? Not likely. More protein? Not likely. The answer would be less ATP, because if you reduce the amount of oxygen, that means this reaction will not go through as well or as efficiently, and then ATP will go down. Alrighty. Number 17. Wow, this is a long test. You guys must have a mean teacher. So but this was another one that we had on our practice. So you guys should have gotten this one right. The sweet taste of freshly picked corn is due to the high sugar content in the kernels. Right? Sugar is glucose or monosaccharides. There's an enzyme that converts the sugar to starch within 1 day of picking. So to preserve its sweetness, freshly picked corn is immersed in boiling water for a few minutes and then cooled. Which statement most likely explains why the boiled corn kernels remain sweet? Boiling destroys sugar molecules so that it cannot be converted to starch. Well, sugar is sweet, right? So if we're destroying the sugar molecules, it would actually get less sweet. Boiling kills a fungus on the corn that is needed to convert sugar to starch. Again, here we're talking about removing the sugar, and we didn't talk about fungus at all. There's no evidence that there's a fungus on there. Plus, we're talking about enzymes. You know what heat does to enzymes, right? Boiling activates the enzyme that converts amino acids to sugar. Well, amino acids do not get converted to sugar. Amino acids get linked together to form proteins, and they get digested back down to amino acids. So boiling does not activate. There is no enzyme that converts amino acids to sugar. These are two completely separate things. Boiling deactivates the enzyme responsible for converting sugar to starch. That makes more sense, right? It says here, enzyme action converts sugar to starch within 1 day of picking. So boiling would deactivate or denature that enzyme. And so the sugar that would normally be converted to starch would stay as sugar and it would be nice and sweet. Number 18. The diagram below represents a cell organelle involved in the transfer of energy from organic compounds. What organelle is this? It's the mitochondria, right? So the arrows in the diagram could represent the release of what? ATP from a chloroplast carrying out photosynthesis. Is ATP released from a chloroplast? No, it's in the mitochondria. What about oxygen from a mitochondrion carrying out photosynthesis? No, where does photosynthesis occur? Photosynthesis occurs in the chloroplast. Glucose from a chloroplast carrying out respiration. Again, chloroplasts do not carry out respiration. That happens in the mitochondria. Carbon dioxide from a mitochondria carrying out respiration. All right, well, we know mitochondria carry out respiration. Those match. And is carbon dioxide a product of that? Yep, glucose plus oxygen produces carbon dioxide and water and ATP. So all of that makes sense. You're going to choose D for that answer. All right, number 19. Which characteristic allows enzymes to function in a specific way? Every enzyme has a specific shape. Enzymes are all about shape. So are hormones and receptors, as you're going to find out in our next units. But enzymes are not made of starch. They're made of protein. Enzymes are not fat. They're made of protein. And some enzymes are made up of 4 subunits, but not all of them. So that's kind of crazy. All right, shape. Number 20. Most of the starch stored in the cells of a potato is composed of molecules that originally entered these cells as what? Starch is a long chain of glucose. I'm not going to write that whole thing out, but starch and glucose, those are 2 words you should always have like paired together in your mind. So starch, when during photosynthesis, plants take carbon dioxide and water from the environment, and together with sun and some enzymes, they produce glucose. Then they link up all those glucose into chains of starch and store them for when more is needed. So starch in cells originally came in as sugars through photosynthesis, the process of photosynthesis. Enzyme, you see the word enzyme, you should think protein. See the word amino acids, you should think protein. See the word minerals. It's really not one of our biological molecules, so the correct answer is B. Number 21. This is kind of like I was just talking about, a sample of food with one particular large molecule. Immediately I'm thinking, okay, it's either protein, carbohydrates, like starch, fats, or nucleic acids. Was treated with a digestive enzyme. Nutrient tests performed on the resulting product showed the presence of simple sugars. What were the original large molecules made of? Well, we know that simple sugars are linked together to create starches, which we eat. So the correct answer is, again, when you think sugars, starches, they all go together. The original large molecules contained in the sample, the original large molecules contained in the sample were molecules of starch. Okay, if it said small molecules after the digestion, then it would be glucose. But the original large molecules were starch. You digest those up, and you get simple sugars back. Okay, 22. Dissolved carbon dioxide in a lake is used directly, but by what? Well, carbon dioxide and water are used for photosynthesis to produce glucose and oxygen. What kind of organism undergoes photosynthesis? Autotroph. Got to know that word. Enzymes have an optimum temperature at which they work best. You should know that the optimum temperature for human enzymes is 37 Celsius or 98.6. That's really the only numbers I need you to memorize for this. It's just useful to know. Okay, so those are just for humans. Temperatures above and below this optimum will decrease activity. Which best shows the effect of temperature on enzyme activity? Well, let's see. You see the above and below the optimum would be where the activity is the best. So below we have lower activity. At a higher temperature we have lower activity. So I'm going to go with B. This would show as temperature increased, enzyme activity decreased. This would show as temperature increased, enzyme activity increased. And this would show that as temperature increased, enzyme activities were first slow, then went through a steep increase, and then kind of leveled off. And we know that enzyme activity always goes up to its optimal temperature and then crashes back down through the process of denaturation. All right, which statement is best supported in the graph below, number 24? The enzymes respond in a similar way to changes in temperature. Well, heck, that sure looks right? They're going up and down, up and down. The enzymes in bacteria function best at 40 degrees. Let's see, which one's the bacteria? This one's the bacteria. Let's see, where is 40? Does that look like it's good? No, that's the lowest possible rate of reaction. That is not a good choice. The enzymes function best at the same temperature. Well, let's see, the human enzyme works best at, looks like 37. And it looks like the bacterial enzyme works best at more like 78-ish. Is that the same temperature? Nope. Do they break down the same substrates? Not a chance. Enzymes only work on one specific type of substrate. They don't work on more than one. They just have one particular favorite. Number 25. Red onion cells undergo the change represented in the diagram. This is most likely caused by the cell being transferred from what? So here you're looking at a plant cell, right? We have a cell wall and the cell membrane. Okay, the pigments are all distributed nice and evenly throughout the cell. It's very, very happy. Then we add salt. Okay, and the salt stays on the surface of the cells. It doesn't go into them. But what it does is it draws the water out to the higher solute concentration. So the water leaves the cells to go to the salt, and that causes this whole thing to shrink up. All of the water's leaving, so it's shriveling up. Okay, the change is caused by the cell being transferred what? Distilled water to starch indicator? That would be silly. We would never do that. Distilled water to salt water? Yep. Salt water to tap water? No. If it was salt water to tap water, it would be the opposite. Although tap water might be different than distilled, but salt water to any fresh water is going to have the opposite effect. So this is distilled water. When you see things shriveled up, it's because we add salt and the water moves to go to the salt. The salt doesn't go in the cell. The water moves out of the cell to go to the salt. Alrighty. Light energy is captured in the process of what? Well, I'm just going to write in things I know. I'm going to write in photosynthesis. We know that. Which stores energy in the bonds of glucose. To be used in the process of respiration. I'm just going to write in what I know first. And then see if that helps me complete the question. All right. A, photosynthesis. Okay, so the only rows that have A, photosynthesis, are row 1 and row 3. All right, now let's see what we have for B. I said glucose. Glucose is organic, right? C6H12O6. I am so bad at this. Okay, that is an organic molecule. Okay, and then because it has carbon and hydrogen. To be used in the process of respiration, I said respiration. Holy smokes, look at that. The answer is 3. Alrighty, so base your answer. Number 27, base your answer to the following questions on the diagram below and on your knowledge of biology. Building block A, building block B, building block C. These all work together to make a protein. What are the building blocks of proteins? The word I'm looking for, and this is really just something that you should have memorized. You should have all the building blocks of all those 4 biological molecules memorized. And if you're not sure about those, look at the slides or look at my review videos. So amino acids. Some of you guys said, 1 or 2 of you guys said this, which actually made me really happy because you said carbon, hydrogen, oxygen, and nitrogen because you know those are the individual things that make up amino acids. But the building blocks themselves, are you really looking for amino acids? I gave credit for this answer because they are made up of carbon, hydrogen, oxygen, and nitrogen. But this is a better answer. All right, we are getting close. All right, number 28. Okay, you guys know this one. So it's all about shape. I put this in capitals for you because when you get further in the question, you have to know that. What I just tell you about human enzymes, what's their best temperature? 37. Keep that in your head. Which molecule would most likely react with the enzyme? Well, here's the enzyme. Let's look at the shape of it. Which one of these would fit in that shape? Because they have to fit together like a lock and a key. That is D. Self-explanatory. And the answer, why would that be? Shape fits. You could be very, very brief. Just say the shape fits or whatever. But it's because if you turn this on its side, it would fit right onto that. Okay, 30. This is where some people got hung up a little bit. All right, we just remembered that this human enzyme likes 37 degrees. Okay, so what would most likely happen to the rate of reaction if the temperature in the solution were increased gradually from 10 to 30? Our optimal temperature is going to be 37. So let's see if we have 0, 30. So if we know our optimal temperature is going to be 37, we'll put the rate over here. You can just kind of picture it. Okay, from 10 to 30, if we know 37 is going to be the high point, okay, and we're going from 10 to 30, we might expect it to look something like that. That's probably like an enzyme curve that we would see for this enzyme. So from 10 to 30, we're going to see an increase in the rate. Some of you guys said that it would start to denature. If we got over 37, this one, you have to know the optimal temperature for this one. If we got over 37, then it's going to start to denature. But up until 37, it's going to be increasing in activity. Alrighty, we are so close to almost being done, and you guys can go enjoy your Thanksgiving. Okay, number 31. All right, some of you guys got stuck on this one too. It's just a different way of representing that cell, that mock cell that we did with the starch in the middle. So whenever you see something like this, we're talking about glucose and starch going through a membrane, try to remember what we saw in our lab. In our lab, we put starch and glucose inside and iodine outside, right? And then after 15 minutes, we saw that we had iodine inside because it reacted with the starch to turn black. And we saw that we had glucose outside because we used Benedict's to test it. And we could still see iodine outside because the color was still yellow. So just think about this. Starch was too big to get out, but the glucose was able to get out. Just draw some more G's in there. Some more S's. Right? Is that right? So yeah, iodine in, glucose in and out. But the starch is going to stay in. Okay. All right, so let's look to see what would happen. Let's draw what would happen to this. Starch is too big to go through the membrane, so all of the starch is still going to be over here. Okay, but if we look at glucose, which I'm going to make red, the glucose is going to diffuse. It's small enough to diffuse across the membrane, but it's not all going to go over, right? You're going to still have some on both sides. It's going to be at an equal balance. It's small enough to diffuse that you're going to have the same amount on this side as you would on this side. In fact, to be really specific, maybe I should be really specific. 1, 2, 3, 4, 5, 6. I should really have 6 on each side. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, right? So it's starting with 12. It really should be equal. 6 on one side, 6 on the other side. I don't think I took off any points for it not being equal. I think I just looked for it being on both sides. But this is what it should look like. Glucose on both sides because it's small enough to diffuse through the membrane, but starch stays over there because it's too big. All right, you guys did a great job on this one. You're going to use this technique so you can add something to the slide without disturbing it. So you're going to put salt. You could say you could use it to put salt on it. You could say you want to put a biological stain on it. You could say you could put distilled water on it. But anything you want to change something about the slide sample without having to disturb it. Number 33, all right, you guys did great on the graph. I don't have those on here. If you have questions on the graph, come see me. The rate between of fermentation and temperature was when temperature went up, the rate of fermentation went up. That's all you had to say. Some of you guys added that it was a direct relationship, which was great. If you look at your graph, I believe you will see that that showed 80, right? I'm not 100% sure because I don't have the graph in front of me, but just take a close look at your graph and see where that matches up. And last but not least, nope, 2 more. Wow, I really did give you guys a long test. Okay, last but not least, one of the effects of Hurricane Katrina, which devastated New Orleans in 2005, was the death of almost all plants in flooded areas. Initially, toxic chemicals and bacteria were suspected as a possible cause, but scientists later determined that the salt concentration in the floodwater caused the plants to die. Picture your onion cells on our slides. What happened when we washed salt water over those onion cells? Right, we just talked about that in one of the previous questions. The water leaves the plant cells to go out to the salt. So those plant cells dehydrate. So we're going to see water moving out of plant cells to go to the surrounding environment. Water does not move into them, right? Water is going to go where there's higher salt concentration. The salt does not move. So whenever you see the salts moving, you just cross that right out, okay? Correct answer is B. Last but not least, number 38, same question. The process of that water moving, it's lovely old osmosis. You could also just say diffusion. I wish this had the text tool so you guys could actually read what I'm writing, but hopefully when you listen, you can see it too. You can hear it too. Okay, so diffusion or osmosis, the water going from a low concentration, high concentration to a low concentration of water. That's osmosis or diffusion. All right, I'm going to stop there. I'll see you guys all soon.